very unique or very nice. Okay. So before I talk about today's topic, I have a simple question to ask you. What is the meaning of, or can you give me another any example for semi-autonomous organelles? Okay, have you heard a term called as semi-autonomous? Yes. Okay, it can be interactive. All right. So just use this emoji so it'll be very easy to understand if you can understand me or not. Okay. So, anyways, so let's get back here. What I was talking about is, yeah, I asked you a question about, do you know or have you heard about anything called semi-autonomous cell organelles? See, there is a story, and the story is very nice. I'll tell you the gist of the story now. See, what happens is when the life was originated on this planet, and the origin of life itself is a great mystery in itself. And if you see the proper scientific evidence for how the life would have originated on this planet, first it starts with chemical evolution and then it goes with biological evolution. So it's chemicals that first evolved. And when the cell or whatever oh, hold on one second. What will happen? Yeah, excuse me, students, just a minute. Mitochondria, yeah, that's right. The chloroplast are the example for semi-autonomous. Okay, mitochondria. Yeah, that's right. That's right, Ankita. Okay, so let's get back. So what I was talking about is the origin of cell. Hold on. Yeah, I hope this is the right. Can you see me properly? Like, can you see the presentation? You can, if you cannot see, wait, I'll just spin it so you can see me in the larger screen. All right. So listen to this. So when the life originated on this planet, as I was saying, first all these magic organic molecules such as RNA, sugars, okay, and even the plasma membrane, okay, it started envelope, okay, it started having an envelope which we call it as a plasma membrane, and within this was like pigments, and it had large amount of RNA, etc. Now what happens is we have two different organisms separately. One is mitochondria, other one is a chloroplast. Okay, so if you look at mitochondria and chloroplast, they are called as semi-autonomous organelles, as some of you have told the answer. So now what happens is, see, these organelles are one of the most longest sustaining symbiotic association. So this is a long term. You might have felt that, but I will make it very short. When I say that it is a very long term symbiotic association, that means that there is a very clear understanding between this mitochondria, chloroplast, and the rest of the cellular organelles. So they have become so integral as such that we think that they are just one. They are just one cell, and it is a part of a cell. No, but if you see the story behind mitochondria and chloroplast, they were separate organisms itself, and they come to fuse or just what do you tell, integrate with the larger cell. So whatever the cell that we see today, we have mitochondria and chloroplast. So they have a unique character. You might have known the reason why they are called as semi-autonomous organelles. One hint I have given you that they were separate organisms and then they got integrated into this cell and then they became a part of a cell. And I have also told you that it's like a beautiful symbiotic relationship that you can come across. Okay. And coming to the larger topic, that is the plastids. What are plastids? Plastids are found in plant cells. We already know that. Plastids are basically anything that contains pigments. Okay, so a membrane bond pigment containing organelles are called as plastids. Based on the type of constituents it has within it, we can classify plastids as chloroplast, chromoplast, and leucoplast. Hold on. Sorry. students, 1 minute. Okay, hold on. Something is wrong with the display, but it's okay. Somebody texted something. Let me check. They have their own genetic material. Yeah, this is a very important point as well. Why having a genetic material makes such difference. Okay, good point. Anyways, so let's go back to the slide. So we have to talk about plastids. Okay, yeah, we have to talk about plastids. So as I told you, the plastids can be divided based upon what they contain. So we have three types of them. One is the chloroplast, chromoplast, and leucoplast. So they're basically found in plant cells. So that's what we discussed. And euglenoids as well. Next one is they are very large and they contain pigments. So the classification of the plastids, as I said, is based on the pigments. First one is we'll talk about the chloroplasts. Okay, chloroplast containing, you know that chloroplast is basically contains chlorophyll pigments, okay, which is a plastid, and also they contain carotenoid pigments. So these pigments, they play a role in as a light harvesting system. So they're responsible for harvesting a specific wavelength of light, which is responsible, which in turn facilitates the process of photosynthesis. All right. So now this is about chloroplast. Now when it comes to chromoplast specifically, chromoplasts, they contain a specific type of pigments, and these pigments are fat soluble pigments. We already have heard about this pigment, that is carotenoid pigments. Same carotenoid pigments are present in chromoplast, but these carotenoid pigments are very large in volumes. So such as carotene, xanthophyll, okay, fucoxanthin, fucoerythrin are some of the examples of xanthophyll pigments. So when I say that it is carotene or xanthophyll, don't think that it is one pigment. Under xanthophyll, you have several varieties of xanthophyll pigments. Similarly, the same goes with carotene as well. Okay, so that is what gives you flowers. It gives you fruits. Okay, the colors of yellow, orange, or red. Okay, etc. That is because of the presence of chromoplast. Now coming to the leucoplasts. Now they are also colorless plastids because their basic function is to act as storage. Okay, so they are known to store several different types of substances, such as you have amyloplasts. It's a leucoplast itself, but it is called as amyloplast because if it stores starch within it, it is called as amyloplasts. Example, we have potato. We have okay, even in the sweet potato, we have it in the roots. Here we have it in the stems. Okay, next one is eleoplasts. So eleoplasts are oils and fats. So for example, groundnut, okay, any of the nuts that are very rich in oils. Next one is alleroplast. Alleroplast are they store proteins. Okay, so these are the examples of leucoplasts. So leucoplasts are what? Leucoplasts are a type of a plastid. So basically, they are colorless and they come in different shapes as well. Okay, and they are categorized based on the type of nutrition they store within them. So we talked about amyloplast, eleoplast, and alleroplasts. Okay. So next one we have to talk about is a very important plastid. So that is the chloroplast. Okay. So now why we talk about chloroplast is we have a significant reason. That is, first one is we talk about the structure of chloroplast. Then next you talk about how the photosynthesis process and the energy production takes place in the plant in anatomy. So obviously you need both of them. So now you're studying anatomy here, and then you'll study physiology there. So that's why it is very important to understand the basic of chloroplast structure. When you look at a chloroplast structure, uh, when it comes to the shape, it comes in different shapes such as spherical shape, disc shape, ribbon shape, okay, or discoid shape, etc. Now, imagine any of these shape. The best one that is very convenient to you is spherical or oval shape. Now, when we imagine any of these shapes, imagine that the shape is made up of two layers and two membranes. We'll call it as the outer membrane and the inner membrane. Okay, these are the two layers. The outer membrane is very continuous and it is quite permeable. Permeable means it allows substances to diffuse or osmosis or transportation happens at a higher rate. When it comes to the inner membrane of the plastic, especially the chloroplast, is basically less permeable compared to that of the outer membrane. Okay, the reasons are very, uh, very simple. The reason why this particular inner layer is not so permeable compared to that of the outer layer is because the inner layer encloses a very important portion of the chloroplast that is called as stroma. So now here the stroma contains a different environment itself, so it needs to isolate that. And this environment is very, very important because it is manufacturing carbohydrates and proteins. So that is why it separates this environment. That makes it very less permeable. Now, as I told you, the inner membrane of the chloroplast, okay, it encloses a uniform matrix that is called as stroma. Okay, so this stroma you can just imagine as a jelly box, a room full of jelly, and the jelly is completely stroma. Okay, in the stroma, what you can see that is a coin-shaped sac-like structures, and these coin-like sac-like structures are called as thylakoids. Now, these thylakoids, how they are arranged is they are arranged in the stacks, okay, one upon another in the form of a large pillars. Okay, so this is called as a grana. Understood? So now what happens is the structure of the thylakoid is very, very important. Okay. Hold on. Is there gap between an inner and the outer membrane? Yes, there is a gap between inner and outer membrane, and this gap is very, very important. And the name of this gap is called as periplastid. Thank you for reminding. Okay, this is called as periplastid play. Periplastid, periplastid space. Now, this space is very, very important, okay, to regulate many of the transportation uh, of the substances across these membranes. And this space between the outer membrane and inner membrane is called as periplastid space. Okay, where was I? Yeah. So yeah, I was talking about the arrangement of thylakoids. The thylakoids are arranged as stack-like structures. Okay, and these thylakoids they have this special type of pigments, which is obviously chlorophyll pigments. But in these chlorophyll pigments, you have something called as quantasomes. So these quantasomes are present in the membrane of the thylakoids. Okay, so now these thylakoids are arranged in a large stack-like structure, which is around 15 to 20 thylakoids are stacked, and these are called as granum. These are called as what? Granum. The pillar-like structures are called as granum. So now, when you observe the structure of a thylakoid, the thylakoid is made up of a membrane. Okay, so the membrane of a thylakoid encloses a very important structure. So that is called as lumen. So I take a disc-like structure, the sac-like structure. What I do is I cut it. So if I cut it, what I can see that there is a very important pulp-like structure, and this pulp-like structure is called as a lumen. And who encloses the lumen? The thylakoid membrane. Okay, which membrane is enclosing it? It is a thylakoid membrane. That means thylakoid has its own membrane. And this membrane is so important for understanding. That is, the lumen of the thylakoid is so important for understanding the process of photosynthesis. That's why you need to remember this name. Okay. Now, if you see the thylakoids, you can see that there is another membrane that is interconnecting thylakoid, that is called as interthylakoid membrane. So basically, this bridge that attaches the two thylakoids are called as stromal lamellae, and the stromal lamellae also participates in the photosynthesis. So the bridge is very, very essential. Understood? So what happens here is when you look at the thylakoids are participating in something called as light reactions, where the photosynthetic reaction that happens in the presence of light. Whereas the homogeneous structure or the matrix that we talked about, that is the stroma of the chloroplast. So in the stroma, what happens is we have very important enzymes, okay, and these enzymes help in the manufacture of carbohydrates as well as proteins. which is very, very important byproducts of photosynthesis. So this is the uh, structure of chloroplast. Now, did you understand? Just give me yes or no. Did the students understand? Yes, no. All right. Others didn't understand. So, okay, I'll move on. If you have any doubts, please put it in the chat box. Okay. So we understood the structure of chloroplast one more time. Chloroplast can come in any shape, such as lens. It can be oval, spherical, discoid, or even ribbon shape. And the length is 5 to 10 micrometer, and 2 to 4 micrometer is the width. So the number varies from each cell. For example, Chlamydomonas, which is a green algae, here it is only one. Whereas when you look at the mesophyll cell in an angiosperm, you will look at 20 to 40 chloroplasts will be present within each mesophyll cell. Okay. So coming to the structure, we know that the chloroplast is made up of double membranes. Outer membrane is more permeable, inner membrane is less permeable. Okay, as you reminded me, there is a space between this outer and inner membrane, and we call this as periplastic space. Within this inner membrane, we have this uniform matrix called as stroma. Within the stroma, we have the coin-like structures called as thylakoid, and they're stacked upon each other to create a structure called as granum. Each of the thylakoid, it has a membrane and it encloses something called as lumen. The thylakoids are interconnected by a bridge made of membrane called as stromal lamellae. Now, the thylakoid and the stromal lamellae, they participate in light reactions of the photosynthesis. Whereas, when we talk about the stroma contains all the enzymes and the necessary things for the manufacturing of carbohydrates and proteins. Basically, they participate in dark reactions. Okay, so that is what is the structure of chloroplast. Apart from these, one of the characteristic feature that makes chloroplast a semi-autonomous organelle is it has a small double-stranded circular DNA, and it has a ribosome of 70S. Now, having the DNA and having the ribosome is like a win-win situation. Why? When we have genetic material such as DNA, in the DNA you have a lot of information that controls the all aspects of life. All the characteristics that you see in the living organisms are controlled by who? By the DNA or the genes that are present in the DNA. Now, how these genes get expressed, or how, for example, when you look at human beings, how come human beings have a certain characteristics, right? So, when you look at this, that is all because of the presence of genes. Now, when we see the genes, how it gets expressed, the DNA tells the cell how to perform a function, and that it does by converting the message that it has. That is, the DNA is converted into an RNA, that is a messenger RNA. So this messenger RNA goes to the cytoplasm, and then the ribosomes come there and they read their message and they construct their protein. Now, in a biological system, 98% of all the functions that you see, biological functions, are controlled by proteins. I've been telling you this. So, mRNA is there, and using this mRNA, see, ribosomes, they can synthesize their own proteins. When you have proteins, you are controlling majority of the life functions. So, since your mitochondria and chloroplast, they have their self-sustaining behavior with respect to genetics, with respect to expression of characteristics, with respect to generating their own energy, okay, expressing their own things. That is why these two organelles are called as semi-autonomous organelles. Okay, did you understand? Yeah. If yes, we will go ahead. If no, you let me know. Did you understand, students? Yes. Okay. So we'll give time for others to sink in and respond. Okay. Next one we have to talk about is ribosomes. Okay. Now, ribosomes are very, very unique organelles. Why they're very unique organelles is because of their composition and their function. It's very unique and they're very, very important. Okay. Now, George Palade first is a person to observe the ribosome in 1953. I think we have discussed a little bit about ribosomes, but yet we will try to do it one more time. I told you that ribosomes are macromolecules. They are not like normal cellular organelles where they have a membrane. Okay, where they have a membrane, and within the membrane you have the constituents of the cellular organelles. It's not like that. The ribosomes do not have any membrane. Now they do not have membrane. Then what is ribosomes made up of? Ribosomes are made up of two important composition. One is the RNA, and it is made up of proteins. Okay. Now, the ribosomes are very, very important, as I've told you, because ribosomes participate in the production of proteins. Okay, what do they do? They form proteins. It's a lag. Okay, so they form proteins. Who forms proteins? Ribosomes form proteins. So they involve with the endoplasmic reticulum and they prepare proteins. Now, how do they do that? When you see the structure of the ribosome, you can see that ribosome can be divided into two important subunits. So we call it as larger subunit and smaller subunit. When you see a larger subunit, they come at 60S and 50S, whereas the smaller subunit comes in 40S and 30S. Okay. Now, this S, we already discussed about the significance of S. S is a sedimentation coefficient, I told you. It talks about the density at which a particular macromolecule separates, okay, with respect to the applied centrifugal force in a centrifugal machine. So that is called as Schwarzschild constant or sedimentation coefficient. I mean, like how fast or how slow you settle down on an applied centrifugal force. Okay, so when that happens, it separates. So usually in eukaryotic cells, you can see that it is made up of 60S, which is a larger subunit, and 40S, which is a smaller subunit. So in total, it becomes 80S ribosome. I told you that it is not additive because maths do not work in one place in cell biology. That is this. Okay. So I've told the reason also. Next one is 50S and 30S, which is mostly found in prokaryotic organisms. Now, the ribosomes, they participate in something called as transcription, where they read the messages from the DNA and they convert it into a protein. Okay, they convert it into protein. Understood? So you will study about the role of how the RNA, how the ribosomes participate in the manufacture of protein in the next class. I mean, like in the next year. Okay. So until then, it's not necessary at this point to remember all those facts. Okay. Here, the next organelle we have to talk about is cilia and flagella. So I think a part of the flagella has been discussed in the earlier part of this chapter, where we were talking about the filament hook and the basal body about the flagella. But now we will talk about it in cross section, where we talk about its electron microscopic images. Now, when we talk about the comparison between the cilia and flagella, they both are very, very similar in their function, in their structure, not in their size. Now, the cilia is like a brush-like structure, which is an extension of the cell wall. Okay, and it comes out, and there are like thousands and thousands of cilia, which are very synchronized with each other. That's why they all, like how we row the boat, the front person and the back person. Okay, we row it together, right? Similarly, all the cilia, they synchronically, they just row themselves, okay, just paddling to the surroundings and then move. Okay, now coming to the flagella, flagella is one to few, many. It's not like thousands or hundreds. So flagella is very, very long and it's a whip-like structure. So basically it rotates, okay, not whips, it rotates. So the entire cell goes into this circular motion and it leads to the movement in the motility in the cells. Both are motile organs which help in cellular movement. So let us look at the cross section, okay, of the flagella. Okay. So when we look at the electron microscopic image of a flagella, what we can see is the flagella shows that the outermost layer of the flagella is a plasma membrane. So within the plasma membrane, what we can see is we can see that there is a radial arrangement of these microtubule doublets. So when I talk about microtubules, so there are two tubules that are involved here, micro, small, tube. That means a tube-like structure, pipe-like structure. Okay, these are microtubules, let's assume. Okay, so microtubules, there are two microtubules. One is A microtubule and B microtubule. So this A microtubule and B microtubule, they are arranged like this together. So this each structure is called as microtubule doublet. So this we have peripheral microtubule doublets. Peripheral means boundary. Across this boundary we have, okay, like this. Okay, I hope it is 9 or 3, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, there is 9, I assume. Okay, so like this, 9 peripheral microtubule doublets are in the arranged in the circular manner around the boundary. Now, if you see here, how do we differentiate between A tubule and the B tubule? Okay, you can see that there are certain projections on the A tubule. We'll talk about it. Okay, now each of these microtubule doublets are interconnected. Okay, they're interconnected by intermicrotubule bridge. Okay, they are interconnected by intermicrotubule bridge. At the core or at the center of the flagella, what we can find is there are two central microtubules which are again interconnected by each other, and they are in turn protected by a central sheath. Okay, so now each of this microtubule doublet, they extend radial spoke towards the center. Okay, radial spoke which is towards the center, which is again made up of proteins. Okay, towards the center and attached to the central sheath. Now look at here, student. Okay, my diagram is very sloppy, but here you can understand. What did I tell you? I told you that the flagella, when you take a cross section, what we can find is you can find that there are doublets. So what kind of doublets we have? We have microtubule doublets. So let's call it as A ring, A microtubule and B microtubule. Now, what we can see is these microtubule doublets are interconnected, correct, right? So they are interconnected. So this interconnections that we can see is called as intermicrotubule bridge. So when I tell that it is intermicrotubule bridge, okay, so that means they are interconnecting each other. Now in the center, what we can see that there are two more microtubules, which is called as central microtubules. So they both are held together and they are protected through by a central sheath. Now coming from each of the microtubule doublets is a radial spoke. So you can see here, they're extending or projecting towards the center, especially towards the central sheath. So this is called as radial spoke. Now, very important things that we have to remember here is if you see the microtubules on one of the rings, you can see that there are some extensions. So these extensions are called as Diane arms. Now, these Diane arms are very helpful for the microtubules. Why? Because they generate the energy that is responsible for the movement of the flagella. Which one? Diane arm, which is present in the microtubule. Understood? Okay. So this entire structure is called as an axone. So as you go down, as you take the cross sections, you can see that the same thing is there everywhere from the tip of the flagella. I mean, like you have the filament, right? From the tip of the filament to the base of the filament, you will get the same structure. Okay. So what is this microtubules? These microtubules are tube-like structures which are made up of special type of protein called as tubulin protein. Okay. So this tubulin protein, they extend downwards in pillar-like fashion that gives the structural integrity to your flagella. Understood? Okay. Yeah. One more thing. I told you that these bridges here they have given as linking protein. These bridges, which are called as intermicrotubular bridges, they are also called as Nixon bridges. Okay, Nixon bridges. So that is because of the protein Nixin is the composition that is made up of a flagella. Okay. Did you guys understand the structure of flagella? Yeah. Students, should I repeat? Yeah. All right. So this is about flagella. Okay. So now, is there a difference between a prokaryotic and eukaryotic flagella? No, they're very, uh, you know, they're structurally quite, functionally same, but structurally quite different. Okay. So this is about flagella. Now, coming to the next cell organelle, and the next cell organelle is centrosomes and centrioles. Very nice organelle to understand and see its functions. So what you can see is the centrosomes are the combination of two basic structures, and these basic structures are called as centrioles. So the centrioles are arranged in perpendicular fashion one upon each other. Okay, so these together they form something called as centrosomes. Centrosomes are very, very nice. Why? Because you see, looking at the cellular perspective, when you look at the cell and when you look at its function, it's so small and it is so well organized, right? For example, let me give an example. For example, if a cell divides and it is going under reproduction or division, cell division, let's say, so one cell divides into two. So when one cell divides into two, now what happens here? My responsibility as a cell when I'm dividing is I have to make sure that my daughter cell also knows what it has to do. That is the information. So this information is with me in the form of genetic material, correct, right? Now, these centrosomes, they make sure that these genetic materials which are duplicated. See, I have bought two copies of the things. My job is to separate these two copies and give it to individual cells. So if I make a mistake in separating these copies, then you know what would happen. Either one cell will end up with high genetic material or low genetic material, which is either way very bad for the cell and it cannot survive. Understood? So these centrosomes, what they do is they majorly participate in the in the cell division. Okay. Second one is they give the cell the ability to move motility. Okay. So that is also brought by centrosomes. Formation of flagella, formation of cilia. Okay, these are all the functions of centrosomes. Understood? Okay. Now, centrosomes, as I told, they are made up of two important structures which are called as centrioles, which are arranged perpendicularly. So centrioles again they are made up of 9 triplets. So you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9. So these 9 microtubules are arranged in the form of triplets. So they are arranged in the cartwheel structure where all these triplets are connected to this central hub, which is made up of a protein core. Okay, so the central hub which is called as a protein core, so which we call it as a hub. Okay, this triplet they are connected to a hub also, we call it as. So these powerful proteins they connect to them. Now what do they do? These centrioles which have these microtubules, okay, they manufacture spindle proteins or tubulin proteins and they form spindle fibers. Okay, they also help in extension of this tubulin in the formation of flagella, etc. Understood? So these are called as centrioles. So centrioles are these triplets of microtubules arranged in cartwheel-like structure, right? So the two centrioles together form this body called as centrosome, okay, which is mostly found in animal cells. Okay. Chalo. Next one, next important structure is nucleus. So there is no need to emphasize the significance of nucleus. We already know that the nucleus is the master center of the cell and it controls all cellular functions. Most all biological functions are mostly controlled by the nucleus of the cell. And what cell does and what cell does not do also is completely regulated by the nucleus. What is something so special about nucleus? Now we know that already we discussed about the prokaryotic nucleus. Now we're discussing about the eukaryotic nucleus. Okay. When we talk about eukaryotic nucleus, we know that U is new and carrion is nucleus. Okay. So the new genetic material, how it is, why we call it as new genetic material? The organization and the way the cell functions completely changes with the way the nuclear material is arranged. So when we look at the nucleus can be visible basically when it is in a non-dividing state. When the cell is non-dividing and the genetic material do not have any work, so they are quite free, and the nucleus is large and it is very visible. Okay. Now, when we see this nucleus, it was first discovered or described by Robert Brown, and then several basic strains were used. Okay, to stain the nucleus. Now we use eosin. Okay, so and chromatin was named by Fleming. Okay, now the number and the position and the shape of the nucleus, it depends. Some of the like red blood cells, they're enucleated. In the beginning, they'll have nucleus. Gradually, they lose nucleus. Okay, they become anucleated cells. Now, when we talk about some cells, they are multinucleated cells, they are uninucleated cells, or anucleated cells, or enucleated cells. So it depends on cell also. Now, coming to the nucleus, as I told, the nucleus is made up of two membranes, so which we call it as nuclear membrane. Okay, so there is outer membrane and the inner membrane. Again, the outer membrane and the inner membrane is separated by a space called as perinuclear space. Okay, so again, it wait very perinuclear space. Now this space, the perinuclear space, okay, is some at one point is punctured, okay, and it creates this pore or openings. So these pores are called as nuclear pores, which are very important. Okay. Now, what happens? What is there within this nuclear membrane? Within this inner nuclear membrane, what we find is a uniform structure called as nuclear matrix. Okay, or we can also call it as nuclear matrix. Okay, that is very simple. So, or nucleoplasm. Yeah, this nuclear matrix or nucleoplasm. Within this nuclear matrix or nucleoplasm, what you can see is there is a condensed spherical shaped structures which is made up of chromatin fibers, and this condensed structure where you have a lot of chromatin material which is in the spherical shape concentrated. So this is called as nucleolus. So outside where everything is uniform, that is called as nucleoplasm or nuclear matrix, and within this nuclear matrix you have where your genetic material is condensed, it is made up of nucleolus. Now, here, in the nucleolus, we know that it is made up of chromatin fibers. Now, when we talk about genetic material, why did we end up at chromatin fibers? See, what happens is prokaryotes, they are so advanced, they know how to very cleanly pack their genetic material. So that packing of genetic material is done by a specialized proteins called as histone proteins. Okay. Now, what happens is our cell uses the very basic. Okay, hold on 1 second. Give me a second, students. Yeah, you can see me, right, students? Yeah. So, where was I? Hold on. Yeah, I think I was talking about the packing of genetic material. Yes. So histones are positive. Our cell uses this, charges of the molecules to pack it. So our DNA is negatively charged. Histone proteins and non-histone proteins are positively charged. So the negative and positive, they attract each other. So around each balls of histomer, so let's say that there is balls of histomer, and each ball of histomer, they can wind up to 150 base pairs 150 base pairs of DNA. Not so complicated. Let's say that your DNA is made up of A, T, G, G, C, C. So you know that these are called as nucleotides, which we discussed in biochemistry. like this 150. So each of this 150, they go like this, and they look like the pearls on the string. So this is called as nucleosome. Okay, this nucleosome is further condensed by these powerful proteins called as chromatin fibers. So that's when you end up with chromatin structure. Understood? So that is how the DNA is very well packed within the nucleus. And in the nuclear matrix or nuclear plasm, what happens is there are very important proteins and ribosomes, and it is a very huge site for the manufacture. Nucleolus is a very huge site for the manufacturing of RNA. So the RNA transportation should happen here. So it basically maintains this entire nuclear environment. Okay. So example for mammalian RBC and sieve tubes, they don't have nucleus, as I've mentioned. And some cells have many nucleus, some cells have no nucleus or one nucleus. Right? So this we discussed. Next one, yeah, I think we have to, okay, we'll discuss this. Next one is the chromosomes. As I told you, as we keep on just condensing the structure of chromatin, we get into this X-shaped bodies called as chromosomes. Chroma means color. Colored bodies called as chromosomes because they appear color under different stains. Okay. Now, these chromosomes are very specific in their number of chromosomes. For example, humans have 46 chromosomes in their diploid state. Okay, and 23 chromosomes in their gametic state, like sperms and ova, they have 23 chromosomes or half the number of chromosomes. So the number of chromosomes define the genetic volume of an organism, okay, and it contains genes and other non-functioning genes also, okay. Now this chromosome, which is an X-shaped body, is basically, you can see that it is made up of arms. So these are called as sister arms. So the sister arms are connected by this structure called as centromere. This position of the centromere, based on the positive position of the centromere, you can differentiate the different chromosomes. Understood? Now, this chromosome, for example, if the centromere is positioned in the center, it is called as metacentric. If it is at the little bit away from the center, it is called as submetacentric. Okay, almost at the corner, it is called as acrocentric. At the end, it is called as telocentric. Okay, so based on the position of the centrosome. Now, if you look at the faces of the centrosome, you can see that there are like disc-shaped structures on the centrosome, which are called as kinetochores. Okay, they are called as kinetochores. So this disc-shaped structure, which we call it as kinetochores, are powerful proteins. So whenever the chromosomes have to be separated, which are the genetic materials eventually, from one cell to another cell, so these kinetochores help in attachment of the spindle fiber proteins, okay, or spindle proteins or spindle fibers. So they come and attach to these kinetochores and they get pulled. Okay, so this kinetochore protein motors is what help them to move from the center to the poles of the cells during cell division. Understood? So sometimes when you look at the X-ray of the chromosomes, what you can see is some regions of the chromosome is very light and some regions of the chromosome is very dark. So that is because the way the DNA is packed within the chromosome. If it is very tightly packed, it is called as heterochromatin. Okay, I'll write it here. It is called as heterochromatin. Okay, and if it is loosely packed, it is called as euchromatin. Since it is very tightly packed, heterochromatin DNA will not participate in the genetic information or giving characteristics, whereas euchromatin is loosely packed DNA, which is a functional DNA where it participates in giving characters to an organism. Understood? So this is about the structure of chromosomes. So they're usually paired structure whenever it comes to vegetative cells or any, for example, skin, brain, or any cells that you take except for your sperm and ova. All of them are diploid and all of them have paired structures. Okay, and this is a very generic structure of chromosome. We'll study about it in detail in the coming year. We have a lot of molecular biology to talk about. Understood, students? Okay, I will end the class here today now, and we will take up the small topics that are left in this chapter. Okay, we'll discuss about it and go ahead. If you have any doubts, please put up a message. I'll be here. And if you do not have the doubts, you can leave the call. Okay, have the next year. Thank you. Thank you, students. Bye bye. See you tomorrow.